Dear students, in the last classes, we have talked about the various types of diseases, how they affect the health of the body. Now in this lesson, what is called immunology, we have to talk about how our body is protected from various diseases and infections. And we have a system, what is called the immune system. Through the effective mechanism of the immune system, we have developed what is called the immune power, the resistance power against various diseases. And that is the system we have to talk about. As Hippocrates, father of medicine, aptly said, you see, we have natural forces within us. So the natural forces within us, nothing but the immune system, are the true healers of the disease. That was the notation given by Hippocrates, father of medicine. So we have natural forces, nothing but the immune system present in the body. If we are keeping well the immune system, we can protect our body against the diseases. Now what is immunology? The study of immune system. That is called immunology, simply we can see. Now what is its function? It gives protection to an individual from various infective agents, the disease-causing agents. That means what's the meaning? It refers to all those mechanisms used by the body for protection from various environmental agents, that are foreign to the body. Now, what do you mean by environmental agents? So it may be in the form of microorganisms or their products, even the food, the drugs and chemicals. These are all called environmental agents. Mainly the microorganisms, the toxic products, the food items, the drugs, the chemicals, even the pollen grains also. Because pollen grains infection causes, hay fever, a type of fever. And those agents which are foreign to the body, what's the meaning to foreign? Any substance which will not be accepted by the body is called as a foreign substance. Even in the case of tablet, the sweet coated tablet, the sweet coating is foreign, not accepted by all. It causes some damage to the body. Even the pollen grain, if it causes a disease, it is called as a foreign agent. Or we have any other type, chemicals, anything else, which are not accepted by the body, they are called as actually the foreign substances. That is the definition for actually immunology. What is immunology? What does it refer to? Now, we have, that is normally immunity power, we can say very often, or body resistance, otherwise called as body resistance. And that is called immunity. The resistance of the body against specific diseases is called immunity. So we can see what is immunity. The overall ability of an organism against the disease-causing pathogen. Simply we can see the overall ability of an organism against the disease-causing pathogen is called immunity. The word immunity is derived from Latin. That is immunis. The meaning for that word, freedom or exempt. Freedom or exempt, that is the meaning for immunities. So that is called immunity. So the ability of the body against various infectious diseases is called immunity. It is also called body resistance. If a person is lacking immunity, he is called susceptible to a disease. So lacking resistance to disease is called susceptibility. This is nothing but lacking resistance to disease. That is called susceptibility. And normally the immune system is highly specific in working against what is called the pathogens, giving resistance to the body. We'll discuss one by one in this lesson. Let's continue further. Now, many immune responses normally do the following functions. Now the many reactions we can see, what is called the immune reactions. Number one, they do what? Number one, initiate destruction. Initiate destruction of the organisms, the foreign organisms, anything else. And two, elimination of invading organisms and also their products. Their products are always highly toxic, so they should be eliminated. So they are also responsible for the elimination of invading organisms as well as the toxic products released by these microorganisms. These are the two important results, the consequences of what we call that is immune responses. One, initiate the destruction. Two, elimination of organisms and their toxic products. Normally all immune responses are destructive in nature, but these responses or reactions are made only in response to those molecules which are foreign to the body. They are not acting against the self molecules generally, mostly. They are always affecting only the foreign molecules. The immune responses are generated only when there is an entry of foreign substance. So they are not acting against what we call the self molecules, namely one's body's host cells. So that's why I mentioned here immune responses are destructive in nature and are made only in response to those molecules that are foreign to the body when they are not accepted by the body and not those of the host itself. This is normally happening. Now, what is happening? This ability to differentiate the self molecules and what we call the foreign molecules is called differentiation between self and non-self. So it is one of the important properties of the immune system. The immune system normally has four important properties. One, memory. Two, differentiation between self and non-self. 3. Specificity, and 4. Diversity. These are the four important mechanisms or four important what we call the properties of the immune system. The immunization and vaccination depends on one of the properties of the immune system, namely the memory. We will discuss later. So the four properties: memory, differentiation between self and non-self. Number 3, what we call the specificity, and the last one, diversity. So this ability, what I mentioned, able to differentiate self and non-self, is one of the fundamental property of the immune system. Out of the four, it is one of the fundamental property. Sometimes what will happen? Now this immune system is failed to make distinction between self and non-self. And so that what is happening? It causes a rapid destruction. So a severe destruction against the host molecules. What we have the host molecules, nothing but the host having this immune system. That results in one disease, a condition called autoimmune disease. What do you mean by autoimmune disease? In autoimmune disease, the immune system of one's body failed to make distinction between self and non-self. So, it is more destructive against the self molecules, leading to a condition, a disease called autoimmune disease. We have a number of autoimmune diseases in our body. For example, multiple sclerosis, rheumatoid arthritis, myasthenia gravis, and so on. So many just actually autoimmune diseases. What is autoimmune diseases? When the immune system fails to attack the foreign body, but attacking its own molecules, self cells, then the disease is called autoimmune disease. We'll talk about later about this one. What is autoimmune disease? Okay, suppose you want, I can write also the board now. What is autoimmune response? I already just described. So I mentioned already one of the important properties of the immune system, self and non-self distinction. Occasionally, I written here already mentioned, the immune system failed to make distinction between self and non-self. As a result, what is happening, and reacts destructively, dangerously against the host molecules and cells, resulting in a disease called autoimmune disease. Autoimmune disease or autoimmune diseases. We have a number of examples. I have given only one example, talk about more later if it is given in the book. Rheumatoid arthritis, a type of arthritis what is studied in 11th standard, is an example for autoimmune disease. Now, we have to go for the next one. What is an antigen? Almost all the macromolecules like proteins, polysaccharides, all the carbohydrates we can see, there is a nucleic acids which are foreign to the body, can have the ability of inducing the immune response in the recipient organism when they are foreign to the body. Almost all the macromolecules, and also we have the synthetic chemicals too. Synthetic organic molecules also can induce what we have normally, the immune response in the case of recipient's body. So, such substances normally we can say foreign substances. Any substance which can stimulate, that is what's called the immune response in the host body, is called a foreign substance. We have examples of the nucleic acids, polysaccharides and proteins. Now we have to go for what is an antigen. Any substance which can elicit immune response is called as an antigen. I mentioned already, it may be microorganism, the toxic product, or any just the macromolecules. We have the chemicals, the food, the color grains, all considered as an antigen. So any substance which can elicit immune response in the host body is called as an antigen. See, the antigen nature is different in different organisms. That's different. Even in the same organism, the nature of the antigen is different. So, even the protein of one's body, while we are doing what is called the grafting, transplantation, cannot be accepted. This is because one's own body antigen is different from another. That's why in graft rejection we have to make what is called histocompatibility for antigens. Whether the antigens are matching between the two persons, the donor and the recipient in transplantation. Okay. Now coming back to this one, antigen. Any substance capable of eliciting immune response is called an antigen. We'll study more about later also. The chemical nature is different for just actually for different antigens. Now, we have to go for the different types of what is called immunity. Okay, I'll take that one later now. So I mentioned about the definition of antigen. Now I'm writing on the board. Any substance that is capable of eliciting an immune response is called as an antigen. I mentioned already the different types of macromolecules, the microorganisms, the toxic products, all are capable of just acting as an antigen. Now let's pass on to the types of immunity. There are two major types of immunity. One is called innate immunity, another one is called adaptive immunity. There is no further classification for innate immunity, but for adaptive immunity we have again two different types and further types, artificial, that is natural, active, passive. We'll discuss later. Now the innate immunity. So it's also called natural immunity, also called non-specific immunity. What is the meaning for innate? Right from birth. Why is it called natural immunity? Present naturally in the body, not actually induced artificially, non-specific, because it is normally just working against a large number of potentially infectious agents indiscriminately. They are not selecting. Any foreign body that enters the body are being attacked. So attacking a large number of potentially infectious organisms, hence called non-specific. What is natural immunity? So it refers to the natural phenomenon of resistance. Natural phenomenon of resistance to infection that an individual possesses right from the birth. So we have some immunity power against infections. For example, the presence of hydrochloric acid in the stomach kills the microorganisms, the skin, or we have the mucus layer, the various chemicals in the form of interferons, complementary proteins, etc. Or we have chemical alarm signals. So those mechanisms present already in the body, giving protection against what we have the infections, constitute innate immunity. So that's why I say it refers to what we call the natural phenomenon of resistance to infection that an individual possesses right from the birth. That's why it's called as innate immunity, natural immunity. I mentioned only non-specific because it can attack any type of potentially infectious organisms without any distinction. There is no specificity, not selecting the individuals. They can attack anybody that enter into the body without any distinction. That's why it's called non-specific immunity. Now, we have a number of innate immunity mechanisms in our body. They are operative non-specifically to attack or against a number of microorganisms. Now let's see what are the different mechanisms or the types of innate immunity, types and mechanisms of innate immunity. They are being used against what we call the infectious agents found naturally in the body. And we'll discuss about the types of adaptive immunity later. So why is it called as adaptive immunity? It is adaptive. The body is adapted once it is adapted to the immune system, so it is resisting against the disease. It's being acquired only after birth. That's why it's called as acquired immunity. Specific. It cannot attack all the organisms. For example, if there is any antibody which will attack tuberculosis bacterium, will attack only tuberculosis bacterium. It will not attack the poliovirus. That is called specificity. So they can recognize specific microorganisms and attacking them. That's why it's called specific immunity, not attacking all the people. Okay, so innate immunity normally constitutes the first line of defense, we can say. We have many lines of defense in the body. It is one line of defense, the first line of defense. We will discuss later about this one. Now we have to go for the types and mechanisms of innate immunity. Now let's see the types and mechanisms of innate immunity are, generally we can say innate immunity includes 4 different types of barriers. They are called anatomical barriers, physiological barriers, phagocytic barriers, and also inflammatory barriers. I'll take one by one. Now anatomical barriers, and which we have 2 examples. One skin, another one mucous membrane lining the digestive tract, respiratory tract, etc. Now what is the role of skin? Number 1, it prevents the entry of microorganisms into the body. It prevents the entry of microorganisms into the body as acting as a barrier. Secondly, its surface has some acidic medium, the pH ranging from 3 to 5. That one suppresses or inhibits the growth. One prevents entry, number 2 inhibits the growth. That's the role played by skin as an anatomical barrier. Anatomy, you know the various structures. Now the mucous membrane, lining mainly the respiratory tract, preventing the respiratory infections, and also in other parts of the body, the digestive tract, urinary tract, etc. What's its actually function? So as it is sticky in nature, it entraps the microorganisms that enter into the respiratory tract and propel out with the help of ciliary movement lining the respiratory tract, entrapping the microorganisms. Also it competes with microbes for attachment, preventing actually just that is the microbes for attachment to the various organs. And normally once they are being entrapped, they are being attached to, that is what's called the mucous layer. And the mucous layer is being propelled out by the streaming movement and also the ciliary movement of the respiratory tract. So it competes with microbes for attachment. These are the 2 organs concerned with anatomical barriers. Now let's go for the physiological barriers. Now the second type of barriers, we have physiological barriers which provide innate immunity to the body. What are they? So the physiological barriers include number 1 temperature, number 2 the chemical mediators, and number 3 the low pH. Now the temperature. The normal temperature of 98.4 degree Fahrenheit normally just to prevent the growth of microorganisms. Normally it's there. And also The fever response is normally a defense mechanism to fight against a microorganism to prevent its growth. So, temperature is responsible for inhibiting the growth under normal conditions, and also at the time of fever, it inhibits the growth of microorganisms. That's the role of temperature. Now, chemical mediators. We have a number of secretions inside the body. They afford immunity to the body. Number one, lysosome. Lysosome is an antibacterial agent. Antibacterial agent. This is one important main question. Antibacterial agent. Now it is working against bacteria. How? By dissolving, cleaving means breaking the cell wall. By breaking the cell wall, that is a protective covering for the bacteria. Now it is able to kill the bacteria. The lysosome, the secretion is normally formed in the saliva, in the tears. We have various secretions of the intestinal juice. Okay, there is an antibacterial agent killing the bacteria by dissolving or cleaving the cell wall, working against bacteria. The next one, interferons. These interferons are cytokines. Example for cytokines. Interferons are cytokines. What are cytokines? Small soluble molecules produced by the cells in response to stimulus between the cells. Now they are acting as messenger molecules, we can say. So they are produced in response to stimulus between the cells, chemical messages. It includes a number of what is called cytokines. For example, interleukins, what I told you in the last class, something about. And also interferons. Now the interferons are glycoproteins. They are working against, so it's an example for cytokine. Then it is a glycoprotein chemically. I am talking about what is called interferon. Glycoprotein. It's working against viruses. That's why it's called antiviral protein. The previous one, lysosome. Sorry, I made a mistake. This is not lysosome, lysozyme. This is not lysosome, this is lysozyme. Lysosome is nothing but the seasonal banks of the cell, the organ. This is a lysozyme. Okay, made a mistake. Correct it. So, the cytokines, there is interferon, one example. It's a glycoprotein. It's working against what is called the virus. It's a protein here is called antiviral protein. Whereas the lysozyme present the saliva, the tears and other secretions, killing the bacteria by breaking the cell wall. So that is an antibacterial agent. Now the interferons were first reported by two people, XII and Lindemann. Two people, XII and Lindemann. In 1957. So the interferons were first reported by XII and Lindemann in 1957. There are three types of interferons. They are alpha interferons, beta interferons and gamma. Simply we can see INF alpha, INF beta, then INF gamma. So these alpha interferons are produced by what is called the leukocytes. Produced by the leukocytes. And beta interferons are produced by fibroblasts. And this is produced by the T cells. So these are the three types of antiviral proteins, namely interferons. Now these interferons are produced by the virus infected cells. Once they are infected, particularly for example the WBCs, they produce this interferon. The interferons are acting as chemical messengers or chemical molecules or signaling molecules. They are just transferring the message to the adjacent cells, which are unaffected. You are going to be attacked, be ready, just face it. That's the information. So, the interferons normally induce, that is normally unaffected cells, ready to face what become the danger. So, Interferons are produced in response to viral infection and protect the cells which are nearby or which are in the vicinity against viral infections. That's about interferons. 3 types. Then what is called antiviral proteins. And this is lysozyme. Once again I am telling you, lysozyme, not lysosome. Now coming to the third under the physiological barrier is the low pH. If you are taking the stomach, the gastric secretion, the gastric juice, which is acidic in nature, the pH is ranging from 1.5 to 2.5, strongly acidic. Such a strong acidic medium normally kills those bacteria and other microorganisms that are being ingested while feeding. So acidic gastric secretion, particularly the hydrochloric acid present in that one, a strong pH, kills the ingested microbes. So these 3 are coming under the chemical mediators. The chemical mediators won't become the 4 I mentioned about, just actually 3, that is lysozyme, interferons and complementary substance. Now the third one under this physiological barrier is the low pH. Under the physiological barriers, I have discussed number 1 the temperature, number 2 the chemical mediators, and number 3 the low pH. Under the chemical mediators we have number 1 lysozyme, number 2 interferons, and number 3 the complementary substances. These are all enzymatic proteins mainly produced by the WBCs. Their main function is to mediate phagocytosis. That means it facilitates the phagocytosis, making the cells to eat. The process of cell eating is called phagocytosis. So the microbes are being engulfed by some cells, what we can say the WBCs, and that is being stimulated by means of these complementary substances formed in the blood. That is what we call actually the chemical mediators, the third one, and the C what I mentioned about the low pH under the physiological barriers. Now the next one, the third barrier, phagocytic barriers. It is affected by a special type of cells, nothing but the WBCs, like neutrophils and also monocytes. The functional monocytes are called macrophages. The difference between monocyte and macrophage, and both are WBCs, monocytes under normal conditions, at the time of functions, it is being converted into a large cell, a macrophage. So a modified monocyte is called macrophage. It is having what is called free radicals and bacteriolytic enzymes to destroy the invading microorganisms. So the main function of the phagocytic barriers by neutrophils, monocytes and macrophages, they phagocytose the microbes, engulfing and also digesting the entire microorganism. So that's about what we have the phagocytic barriers. Even the neutrophils and macrophages, these two can just leave the blood vessels entering into the intracellular spaces where the interstitial fluid is present. So they are called as wandering phagocytes. Wandering, always moving here and there, giving protection to the body, hence called wandering phagocytes. We have, that is, the macrophages, or simply we can say the neutrophils and monocytes are called wandering phagocytes. They are invading all parts of the body, killing the microbes, engulfing the microbes and digesting the microbes, hence called wandering phagocytes. That's about what we have as phagocytic barriers. One more barrier, what we call the inflammatory barriers, the last one under the innate immunity. The last barrier related to the innate immunity is inflammatory barrier. So we have the last one, inflammatory barriers. So you normally see that one at the time of injury or at the site of injury or due to what is called infection, there develop, we have redness, swelling, pain and generation of fever. All these indications, the symptoms together cause inflammatory response. This is because of release of certain chemicals. They are called chemical alarm signals or chemotactic signals. They are called chemotactic signals or chemical alarm signals that resulted in the following responses together called inflammatory response, redness, swelling, pain and generation of fever. So at the place of tissue damage or due to infection, these two factors induce the leakage of vascular fluid, nothing but the blood, from the blood vessels into the spaces where we have the infection or injury occurs. And this fluid contains, I mentioned already, the chemical alarm signals or chemotactic signals. What are they? Number one, histamine, the one which is involved in inflammatory reactions. Number two, serotonin. Number three, prostaglandins. Now the histamine is secreted by the mast cells, one type of what is called connective tissue cell, mast cell, MAST. And that one actually produces the histamine. So these chemicals cause an influx of what is called actually attracting, influx is referring to just coming to the place where you have the infection, accumulation of what is called the phagocytes. And these chemicals cause the influx of phagocytes to the affected area where you have the pathogens are present. So the phagocytes are moving towards that area because of the release of chemicals, and that one just resulted in destroying the invaded microorganism. The process of leaving the phagocytes from the blood vessels to the intercellular space, or we have the leakage place, is called diapedesis. This is nothing but the squeezing of WBCs to pass through the wall of the blood vessels to reach what we have, the tissue fluid, or we have that is known as the affected area where we have the leakage of the fluids. This is simply the squeezing of what we have, the WBCs, through the wall of the blood capillaries. And that was first reported by, that is Meschinikov. That's the name of the person who observed diapedesis of WBCs. Okay, so what is inflammatory response? What are actually the inflammatory barriers? Nothing but actually the various chemicals that cause the influx of what is called the phagocytes towards the affected area so that they can kill and destroy and also just swallow or digest the microorganisms.